Yeah, thank you very, very much. Also, good morning, good afternoon, and good evening around the globe. And I'm located in Switzerland, and I will use the next 15 minutes or so to talk about collections and preservations and needs for microbiome research. I think you can see the slides. And so let me go back one slide here. So,
all good.
okay, very good. Okay, I have three important take-home messages, and I want to start with them right away. So first of all, an important aspect is that collections are variables. They serve different purposes and different future uses, and therefore every collection has its unique aspects and requirements. So what is very, very important is that you try, if you set up a collection, that you try to document the context and the methods, because this is very important to increase the overall quality of the collection. As simple as it sounds, that's a very, very important take-home message. Then also preservations are very variable. There are many different standards. There is not the best standard which fulfills every need. It really depends on your setup, on your research question, what kind of preservation method you should take. And I think a very important aspect is for both collections and preservations, if you're planning a study, stick with one standard in one project. So do not change the standard during the project duration so that you do not introduce biases down the road. Now, obviously, there's a large need for documentation in this regard. And if you document well, this is really a key aspect to ensure interoperability within your own research endeavor, but also to actually compare your data to other people generating microbiome-related research. And to do this, there are more and more professional software tools around. And one type of software tool I will also briefly introduce you to is the so-called Biobank Information Management System, a BIMS. And this can help to really professionalize your documentation. So I think we are all aware what biobanks are, but microbiological biobanks have a few specific characteristics. I mean, they clearly serve also the progress of biomedical research and development and potentially also therapies. The shared characteristics are that biological material is used in a standardized form. And this standardization, I really cannot repeat myself more often. This is a very, very critical element that collections are done in a standardized way. The data, which can be linked to a particular sample, can be very rich and very diverse. I just point here out one example. So antibiotic resistance of a single strain. Now, antibiotic resistance, you may think this is a static field and a static value you measure, but criteria to interpret resistance are changing all the time. So if you follow UCAST as a guideline, every year a new guideline comes out, and it can be that the category for resistance and susceptibility is changing. So this needs to be reflected in the data. Versioning is a very, very important keyword in this regard. And then obviously you should also be aware that future usage of stored material is also quite diverse. If you have single strains in your biobank, well, that's maybe not the topic of this particular conference, but single strains, that's a potential infinite source because bacteria, you can regrow them, but it's certainly limited if it's a complex microbiota. Maybe in the future, we will be able to make mixtures of single strains and then generate the microbiota. But I think for the moment, microbiota samples are quite limited and in terms of how often you can use them. Now, two typical examples of microbiological biobanks would be a population-based biobank and a disease-based biobank. In terms of a population-based biobank, this could be a cross-sectional study, let's say from nasopharyngeal swabs from your medical students, and they would serve as a healthy control group, let's say. And obviously you have certain aspects you want to describe in such a healthy control group. If it's a disease-based biobank, it might really raise also some questions regarding the biosafety, and you should document if there are certain pathogenic microorganisms in such a biobank. So when you set up a biobank, the primary process of sample processing and setting up the biobank is very important to kind of lay out. It all starts with the sample recruitment. This can be a patient, but it could also be obviously an environment or an animal. Then you generate some data from that sample. So it could be a clinical assessment where you describe the phenotype of the patient. It could be microbiological characteristics like the resistance profile I've just mentioned. It could also be genome sequences of the microbiome. And this data has to be stored in a certain structured way. And this is normally done in a so-called data warehouse. And this data is linked with the biobank. So that's the storage process. And obviously, at a certain point, you will also access why do you bank stuff? Because you also want to reuse it in the future. So it's obvious that you also have ordering requests and shipment requests, for example. And that's a typical flow of a sample coming in and a sample leaving again the biobank. And this has something to do with process management. So when you set up a biobank, make sure that you cover typical use cases. So for example, what happens if a patient decides at a certain point that the person does no longer want to be part of your biobank? You need to have an established use case, how you can take out samples and really delete the information of the patient. So it's very good if you think about typical use cases while building this up. Documentation is key, I mentioned. So obviously you ask yourself, for what do you store and why do you store it? And different types of material have different information you can store. We generally call this metadata. So it can cover, metadata can cover areas like storage, like associated information, quality, quantity, consent information. And just as an example now, quality could be, for example, how was the sample handled before it was frozen? What kind of DNA extraction kit was used, and so on. I mean, there are many, many examples of what you could actually document in a biobank. Now, how do you do this? How do you manage this kind of information? And I think a first step to do is be aware how rich data can be. And don't use Excel, please. I mean, many people use Excel as a tool to manage their biobanks. And even in Switzerland, we made a recent questionnaire and we found out that about 60% of microbiological biobanks are managed with Excel. But Excel is limited in many ways. And for this, there are more and more much more suitable solutions coming out. And this is what I mentioned in the beginning, the Biobank Information Management Systems, BIMS. So they can handle different types of information. They can manage different use cases, different user roles. And there are commercial solutions. Some of them are quite expensive, others are much more cheaper. There are also open source solutions, which are very, very good. And there's even one which is in between, that's Open Specimen, something between commercial and open source. So how do you select the best BIMS software for your needs? Really think about the requirements. In my center, when we acquired such a software, we were carefully thinking about what kind of information do we want to store. Excuse me. And we came up with 180 requirement items. I'm not listing all 180 here, just that you see the categories. We had IT requirements, data requirements, functional requirements, regulatory, patient, and non-functional requirements. If I take, for example, functional requirements, we wanted to have a storage inventory management system, label printing, search functions in the biobank, and study management. So these are aspects which a biobank tool should be able to handle. And to set up these list of 180 requirements took us 6 months. So really, it's just an offer. If you're currently requiring such a requirement analysis, drop me an email and I'm happy to share this list with you. What you should also do is test the company you want to buy the software from. So you can send them easily a legacy strain collection, samples you have already in your Excel sheet, and tell them, please import the data into the software. And then you see how good a company actually performs. And I do not have stocks. I'm not affiliated with this company, but we decided to buy Open Specimen. Open Specimen is a very interesting tool. It's a completely open source software, which you can download from GitHub and use it. But there's a company called GloMiNe, which customizes Open Specimen for you in your institution. And this could be a very interesting solution because you have a lot of community inputs coming from the GitHub approach, and GloMiNe is using this input and then commercializes the best solution. So that's a quite nice business concept they have here. Now, why is data and data management so important? Because it has something to do with quality. As you may know, there's a huge crisis of reproducibility in research. And this was very nicely summarized in this PLOS Biology paper, which is a couple of years old now. But in the US, every year, the amount of irreproducible research generates costs of $28 billion every year. And why is it not reproducible? Because there are missing information in preclinical data. So for example, it's not well enough documented what kind of biological reagents was used, what reference material was used. The study design is insufficiently explained. Data analysis and reporting is not good, or laboratory protocols are not standardized. So it's very, very important that samples and sample management is well documented. Now, a way to go is the so-called SPREC code. And SPREC stands for Standard Pre-Analytical Code. And this is an incentive to actually harmonize the sample description in a biobank. And this also works for microbiome samples. So for example, if you have a saliva sample or a stool sample, you can add certain abbreviations. You can process and describe in a very dense way and in a standardized, controlled vocabulary all the steps which were done with that particular sample. So SPREC code is something you should consider when building up a biobank. Now, in the last part of my talk, I will talk about preservation. And here again, the question is, for what do you later store the sample? And based on that particular research question, you can think about how you benchmark the quality of this particular use case. So let's say if you want to reculture the bacteria, you may need to make sure that the bacteria stay alive when you culture them, or you might want to use them for fecal transplantation, and then you all of a sudden have safety issues for the transplant recipient. So you want to, for example, check if there are other harmful pathogens or viruses in it. And that's something which is very important to consider. So benchmarking the quality is a very important point. And I just cite here one study, which is maybe a bit unusual because they used metabolites in the sample as a biomarker for quality. And then it's a lot about standard operating procedures. So processing the samples, freezing the samples, DNA extraction, these are all aspects which can be standardized within a certain study protocol, but there's a huge variability. And I want to show you this variability on two slides. So this is the impact of sampling and DNA extraction methods on stool samples. And I put here a lot of reference, which you can later on read because it's recorded. You should be able to find them. And just to make an example here, this is the DNA yield you get out of a sample in nanograms per microliter. SK1 stands for stool container. SK2, that's a flocked swab, and SK3 is a cotton swab. So the type of sample collection tube you use already has an impact on the amount of DNA you will get out. And QS and PS stands for QIAMP DNA stool mini kit and the PowerLyser Power Soil DNA isolation kit. So obviously the kit also plays a huge impact. That's not very surprising, I guess. So it really matters what you use. And I would highly advise that in the beginning of a project, you do some basic comparison and find out what suits best for you. Because down the road, it has an impact on the OTUs you can generate. You see that there are also some differences, maybe not huge differences, but other studies show much.
Hello, Adrian. Your time is up, please. So kindly wrap up.
Okay.
Thank you.
Okay. Yeah, thanks a lot. I will just quickly draw my conclusions here. Collection and preservation. Ask yourself, what do you want to use the sample for? Standardization is key. There's no single gold standard. Think about strobe metagenomics. Google that. It's a very important standardized way how you can describe information in the in a study. And think about a biobank information management system, so not an Excel sheet. Thank you very much, and I'm happy to take questions if there's still some room.
Thank you so much for your great talk. If anybody has any questions, participants, please write it in the Q&A, and then he will answer it for you. Maria says, which biobanking system do you use in your lab?
Sorry, the biobank information system we use is from Open Specimen. That's the tool I mentioned from Krishagni, and I think it's really it's a reasonably priced and it can do a lot. So it's really a nice one. And as said, if any of the audiences needs a requirement catalog, like what should you look for in detail, because we spent 6 months on putting together all the requirements, and maybe you can reuse that in an efficient way. I'm happy to share this. You can drop me a mail.
Thank you. Another question here says, biobanking tools are often very expensive for research labs. Why do you recommend as open source?
It's again the same. Open Specimen exists as an open software tool. You can download it for free on the provided GitHub link you saw in the proposal. If you, I think the calls are or the talks are recorded, so you should find the slide. But you can also drop me an email if you want to have the GitHub link. I'm happy to share that with you.
Okay, thank you so much for your talk, Adrian.